Okay, so I did a little bit of network analysis for my PhD, but what I use is, so I did gene expression. I don't know if anybody knows, everybody knows what I did. I assume that everybody knows what I did in my PhD. I did gene expression with corals. So corals, we don't have a lot of information about the genes in corals in general and about the functions of these genes. And in particular for soft corals is like all the information is very messy and lacking. So one of the options so you get your gene expression data, and there is a lot of unknowns, like you know which genes are overexpressed or not, but you don't know what these genes are doing. So one of the uses of network analysis for gene expression is to try to find other genes that are related to a gene of interest, and you can kind of basically infer a little bit what will be the function or of that gene. So this is why I started like kind of being interested in this. Because of lack of time, basically, I was not able to dive into WGCNA, that is kind of the package in R that and in bioinformatics that people use to analyze gene expression, but I use another package that is called PETAL. And so the PETAL, basically what it does is that it will create this network from your gene expression data, and it will not ask you to identify any of the parameters that WGCNA is asking you to decide. So in certain point it's good because it's more reproducible. Like you can throw my data many times and you will get exactly the same, exactly the same network. Rather than with WGCNA, if you change any of these thresholds of parameters, you might not have the same results. So that's kind of the tricky bit. Now that I want to publish, I have been advised that it's maybe better to go with WGCNA because PETAL hasn't been used extensively. So it might be rejected just because of the choice of the package. So anyway. And now, as I told you before, I'm just going to go through like a little bit what is network analysis. So if you try to find a definition of what is network analysis, it's very relative. Like nobody gives you like a straight definition. So basically it's the analysis of the graph. So you have some elements that you kind of assume that you can put them together in a network, which means that they have relationship between each others. And then you use this network that you built to basically get information about how these elements work. So network analysis is the study of these graphs, basically. So it can be used in many types of data. Like you only need an adjacent matrix, that is called, but your data itself can be of anything. So just some basics about networks. You have the elements that you connect will be the nodes. So for example, in my case, that were genes, each like the blue circle will be one gene, yellow another gene, and so they will be connected by edges that are the, let me see if I can find the laser. I cannot find the laser. No.
You can see your mouse, I think.
You can see my mouse?
Yeah.
Yeah, Okay, that's cool. But you cannot see my notes here.
No.
But my mouse, yes. Oh, this is cool. Okay, that's fine. So oh, I changed the slide. So anyway, so the connections between nodes that are called edges are, for example, in gene expression, it can be that one gene is co-expressed with another, or it can be if your nodes are people, is that the person A knows person B, or web pages. So how the elements that you use to build your network are connected to each other. Then there are networks that are directed or indirected. Oh no, sorry, this is, yeah. So when they are undirected, they don't have a directionality. As you can see in this, between the yellow and the orange. And directed means that we kind of have a previous knowledge that the orange element is going to affect the blue element. So for example, in biology or in gene expression, it could be that is a transcription factor that you already know that is a transcription factor that will affect your gene. So for directed networks, it's basically everything is the kind of the same. What changes is the adjacent matrix that you use to build this network. And again, this is information. There are some softwares. I don't know much about directed networks because you need very rich data to be able to do a directed network, which means that rich, I mean, you need a lot of prior knowledge to build a directed network. But there are some softwares that if you give them enough information, they kind of have an algorithm that they can infer directionality. Yeah, they can infer directionality, and I don't know which ones they are right now, but I can look for them if you are interested about them. For me, and gene expression is like almost not useful. Like people that use directed networks are like humans that work with humans or mice in very particular data sets that they have a lot of information. Then there are different types of networks that which type of network you use or to build which type of parameter you use to build your network will be reflected in how the graph of this matrix shows, right? So there are like regular networks that, as you can see, is like a very nicely made pattern. All the nodes are connected in the same way. There is this small world network that I will go into more detail later, where there are like all the nodes are, most of the nodes are connected, but it's not a very regular pattern. And then is the random connection between nodes that it doesn't have any structural element, let's say. So in network analysis, it kind of started that people started seeing that networks were built, like let's say relationship between humans and how you know one person or the other. And then the people started like trying to understand why this pattern emerges. So this is the small world type of network. That's how kind of the model was built. So it's called a small world literally because, you know, that expression that we say, oh, it's a small world, like, you know, this person, like a small Townsville, basically, that the smaller the venue, the more people you are interconnected with. And so again, it's just like a type of mathematical graph where most of the nodes are not neighbors to each other, but the neighbors of any given node are likely to be neighbors with each other. So another important element of this small network, small world networks, is that most of the nodes can be reached from another node in very small number of nodes, so of jumps. So for example, I mean, I don't know, you can reach this node here from this one to this one, just going through this one like this, and then jump into this one directly. So if it is a connection, I don't know, like how I met, let's say, Legana. Legana worked, she is Legana, and this is Ira. So Legana worked with Ira, and so that's how, yeah. And so the first person that kind of, I think this is what I have read, maybe I'm wrong, but they is the person that developed these 6-degree distant links. I don't know if you have heard that from any other person, there are 6 degrees of separation between you and that person. And based on that, these smart people, Watson, Strockatz, built a model of the small network. So then, sorry. Basically, most of the natural, I don't know if it is natural, it's a correct term, but natural elements in the world are, if you try to build a connection between them, they will be connected through a small world network. Then there is the distribution of these connections. And so this is when my stats is not like extremely good. So I'm just going to say what I kind of understand. And is that what we need with these small world networks is basically a scale-free, what you find is a scale-free distribution. What you have to allow in your analysis is a scale-free distribution, which means that follows a power law distribution. I think that they are equivalent. And so let me read this, because which means that we will have like a low number of nodes that are highly connected and very many nodes that are low connected. So I try to explain this comparing it with a normal distribution. So here you have the degrees of relatedness and the number of nodes. So in a normal distribution, you will have low number of nodes with low degrees, and also low number of nodes with the majority of nodes with a high degree of interconnectedness. But what a free scale means is mostly this, that you have a very small amount of nodes with high degrees of connection. So there is always like a social butterfly that will know everybody. And so if in social networks, as an example, so if you are if someone new comes to this community, there will be more likely that they will get connected to this person that has high connections with the rest of the network, social network. And then that there is very and then most of the nodes are almost loners. Like, you know, most of the people in the case of a social network will have some connection with other members of the network, but not many. So in this little graph, you kind of can see that there are these hubs that are called, and this is representative of any other type of data that you use to build your network. So it could be genes, genes that are super important and are connected with other elements, or web pages, for example, that Google, it's one of the most visited networks. And then from there, you go through, oh no, my neighbor is drilling. So sorry about that. So anyway, can you hear me still?
Yeah.
Yeah, I'm sorry. Imagine living with that. So does that make sense? I am making sense. Do you understand what I'm saying? Yeah.
Yes.
Okay, if you don't understand, let me know, because I kind of, yeah, anyway. Then these free scale networks have some assumptions. So the first one is that a free scale distribution will allow growth, which means that the number of nodes can increase over time in the model. And then it also assumes preferential attachment, so that more connected nodes are more likely to receive more new links, is what I told you, that if a new person comes to a social network, is more likely to go and meet this person than this one. So this is very, very important if you're going to do network gene expression analysis, because if you don't know, or these assumptions are not real for your biological study, basically the network analysis might not be what you need. So the networks analysis, it's kind of a young field. It started, the popularity increases in the 90s because we started changing from studying like elements to systems, and we started having like high dimensional data with high throughput data in genetics, or geographically as well, and even in the web, in computational information, we started like having these huge amounts of data that people started noticing that the only way to make sense of them was through network analysis. So in biology, we have some specific biological networks. So for example, protein and protein interaction is kind of the first biological field that started with network analysis. And mostly here is like this edge represents when proteins are actually interacting with each other, so they have the information about it. And there is a lot of modeling of protein and pathways that have been done with this type of techniques or of analysis to build, for example, the KEGG pathways, like to build cellular pathways that we now know, and we are like, oh yeah, this one it's interacting with this other one, and this one interacts with the other one. So it's true that there is a lot of manual work that was done, but it was also validated through these mathematical models, which is really cool, because this is like you just throw information to a network, and the network gives you all these patterns of connections. And when the patterns of connections match the biology, is like the holy grail. Then in terms of the same concept of protein interaction can be used for gene expression. And as I explained in the introduction, like basically they are used to find unknown functions of a gene, or to also validate notions or the functions of genes through knockdown. So now there are some ways that you can eliminate one element from your network and see, model how that elimination will affect. So because we have these hubs in the, or important nodes in these small work and scale-free networks, once you strategically eliminate a node that you think that is important, it will have huge consequences in the structure of your network. So if you, let's say, if we translate that to biology and gene expression, if you knock down a gene by eliminating it or from your network, you can visualize what that will do to the cascade of your network, which is kind of nice. And of course, then probably validated with true biology. No, it's not true biology, but with the experimental biology. Then what else is important? So in gene expression and co-expression network analysis, if that's something, a path that a person is taking to investigate their question, it's very important to incorporate from the beginning of the experimental design some elements, and those are that to have a co-expression network that actually can reflect the reality, we need like multiple conditions or treatments or traits. Maybe, so they recommend more than 3 or more. So it is, you can do a network analysis with the treated and control and untreated and control, but you will miss a lot of the elements that the network can actually give you. And also you can have a lot of false interactions or connections, because you are building your network from the data that you are giving the algorithm. So if you give them like a very small amount of data, your network will be, and the connections between these elements will be compromised. So that's one of the most crucial things, and the part that is most criticized about network analysis is that there is not yet like a set of rules, let's say, that are in place for you to use network analysis or not. So a lot of people don't take this into account, and it ends up with false positives. So these multiple conditions will help to give enough information to the program to build your network in a more biological relevant way. And then the other really important element is to have a lot of samples, like to put in your network, because again, is the randomness of the connections and how it's built. It's very likely that you, I don't know, very likely, but it is something that has to be taken with a lot of with a grain of salt, of how many replicates you put and how you interpret your data about 2 genes connected if you have 3 replicates. Then again, like, understand the limitations of the model that you use. So it's true that we are assuming that these genes that have many connections are very important, like many edges to other genes are very important, specifically in co-expression analysis, but that is not biologically necessarily true. So maybe you have a gene that has many connections, but that gene might be actually just very irrelevant. So I think that's one of the other huge issues, that when network analysis is published and these modules of important genes from cancer research to how organs work and everything, like people find that the modules, like the group of genes that they found important, are not reproducible. So if like the connections between genes are not found once another experiment is done in the same system, if that makes sense. So that is like a huge issue, because it means that the whole interaction between your genes is not true, basically. So it's very nice and easy to find these connections, but the necessity of the biological validation is crucial. So if we jump to WGCNA, they have these like very basic work through what the program aims of the package aims to do. So of course, the first thing is to construct the co-expression network, and for that you need the adjacent matrix or the correlation matrix that you build. So this step here and in the WGCNA tutorials is like just announce, or they just say you have to do this, but actually it's very tricky to go through cleaning your data and which data you are going to give to WGCNA or any other program to build, or any other package to build your network, because you have to have, from there you have to decide which elements you want to give to your network, which samples you want to give to your network. So just as an example, there is people that build networks with the treated samples and networks with the control samples, and then they merge the two networks. And so that gives you one type of analysis results, and of course the interpretation of what you're doing. The other option is to just drop everything in the same matrix, like all your treatments and genes in the same matrix, and just see what the network will look like. And ideally, what you want is that both networks are congruent, right? That no matter what you use, your modules, I think I didn't define modules, but modules is like a group of nodes that are interconnected in certain degree, that they are their own little mini network. And so this mini little network might have a biological relevance, or I don't know, in there is also examples of in economic and finance theory of how different enterprises almost govern the whole economy of the world almost. So these little networks or modules need to be consistent despite how you build your network. So because that consistency sometimes fails, that's when the issues with this analysis are kind of raised. So it is very important. So when I do the WGCNA tutorial, I will go through, like, this is why I spent so long and I couldn't finish, because I went through how to build the database, and they give some step by step, but it's more just like how to wrangle your data because you need a certain type of data to build the adjacent matrix, more than like which treatments to use, which samples to use. Do you want to get all your transcriptomic data into a network, or you want to find a threshold of expression? How to eliminate, what does it mean if you don't eliminate certain low express genes for your network and for the following analysis? So after that, that's not what I want. So I did this. Here it is, like the module based analysis. So once you already have this network analysis, WGCNA allows you to make certain clusters, like clustering like a heat map, basically, as you see in the little graph. And then here is the tricky part where you decide the threshold. You decide where to cut the your matrix, or the threshold of how do you say, of how to build your network to the following interpretation of your graph. So this is the first step that we need to make that decision, and that's very tricky and subjective, and it has to kind of be backed up by, or just, it has to be mentioned, let's say, in your analysis, why you did and which threshold you choose for your data to be reproducible. Then after that, you already have your modules that are found with this identifier module or clustering with that use hierarchical clustering. And once you have this group of genes, you need to give them some significance. So WGCNA has the potential to integrate other databases to your analysis, and so you will be able to like give them ontology, which means like what is that gene doing, functionality, and based on that and the clustering of your genes, you can make some pretty graphs to know what your treatment is doing to your genes. This part is also very important, and in terms of corals is very tricky because we don't really have very extensive information about it. So I'm a bit like, I'm always worried about ontology because they give like very big names to your modules or to your group of genes that not necessarily reflect the truth. So this is another step that people needs to be kind of careful. So basically it's a risky analysis. Like if you go through it, like the reviewer can actually like cut you in little pieces. Finally, what they explain here is that you have your big data with all your modules. You already know what these genes do, and it sounds like simple, but you might end up with hundreds of modules and hundreds of these biological connections. So there is tools in that WGCNA allows you to kind of reduce this amount of information to the most important elements of your network, and it does it like kind of with some parameters that you will give, like it can be if the genes have to have certain threshold of expression as well, and have to be connected, overexpressed in certain samples, or I mean, you can play a little bit with this tool to get to this more reducted type of graph, and be able to actually interpret your data. And yeah, so again, there is like this is like more experimental validation and the biometrics, and so biomarkers. So I'm not sure, this part I kind of don't understand what or how WGCNA does that. I think it goes back to your network after you have done all these kind of filtering and manipulation of your data to see if it actually fits in your network. But I'm not quite sure. So when I do the tutorial, I might know more about that. So yeah, so that is it. Like I have some resources here that there are these 2 books of this guy that are very that they're very good. They're a bit expensive, but he writes quite well, and it's easy to read. And then like there are very clever people that have done already workshops of network analysis, and so I put the YouTube videos that once upon a time I saw, and that I use also to try to explain and find the vocabulary to explain what network analysis are. But I mean, be my guest and check like everybody talks right about the same, but they are like the vocabulary is not very in synchrony between all the people that do network analysis. So that makes a bit tricky to explain, and also like to go through the different steps of what you need to do. So yeah, that will be it for me.
Cool. Thanks so much, Natalia.
So if you have, I don't know if it was comprehensive, but if you have any questions, let me know.
Yeah, I have a few, but I'll let other people go first because mine are very uneducated, non-genetic related questions. So
Yeah. The books, are they in the library? I don't know. I don't think so. I tried to, I bought the books, like this one, the first one, during my PhD. And this one, I think this is also like it was published in 2018, I think. And I think that I downloaded from like a Sci-Hub type thing. Let's not put that in the YouTube channel, but yeah. So
sorry to interject. I'm just having a look on the library website. And so
are they there?
The statistical analysis network data with R it's in the Townsend library. And the other one has some online options. Let me see if we have full availability. I'm not sure.
I mean, I remember that I bought it.
Yeah, you can. The full text is available online, so you can access it via the library.
Okay. Yeah. So if anybody works with network analysis and knows more, I will be happy to talk, because I also have a lot of questions. I think it's quite tricky to set up the proper parameters. Yeah, and to interpret the data after that.
Isn't the analysis like available for everyone, you know, no matter the network, whether it's gene expression or, you know, social interactions? It's just, you know, the interpretation of what it means.
I mean, the yeah, the interpretation of what it means depends on what you are, what is the element that you use to build your network. So if it is social interactions, there's a lot of social network analysis. I don't know if you have seen it around, like it's huge, but they have pre knowledge of the stuff that they are trying to build a network on, rather than for us in biological data, you try not to make these assumptions. And so that's what's one of the reasons why I wanted to use network analysis on my PhD, is because I wanted to go kind of blind, just to build a network, see how the genes are interconnected with each other, and see if there is any biological meaning of the network. But I mean, Ira is not here, but there's people that have strong feelings about going blind, because you kind of, in WGCNA, you need to make these assumptions. So yeah, there is like people that work in cancer research. I don't know if someone here works in humans or cancer research, that they do use it to infer function, which is really cool. But I don't know if in a non-model organism we can do that. Yeah.
Yeah.
Mhm.
I was just gonna say it's super cool, and that last part about causality and stuff is probably going to relate to what I'm presenting in 2 weeks about causal models.
Oh yeah, okay.
So we can talk more about it in 2 weeks.
Yeah, maybe, like, I think it's a causal relationship. That's what they call it, like the adjacent matrix and causal relationship.
Yeah.
But if I understand properly, the causal relationship can only be analyzed when you have a directed network.
I think usually, yeah.
Yeah. So yeah, then, well, maybe this is something again that like there are many uses of the network analysis, that you can merge different types of data, not necessarily only because it's just numbers, right? It's just a matrix with relatedness. So if you have, I have seen papers with microbiome data and abundance and things like that, and gene expression, and then they merge to get modules related with changes over time. So actually, that's why I wanted to do this presentation, because I want to try to use network analysis to merge these two types of sets of data.
In terms of the actual analysis, is it like super computationally expensive? Does it take a long time to run?
It depends on how many, let's say, genes you have. It depends on how much data you are putting into it and how complex is your network. So I'm like, the analysis that I did, I tried with all my transcriptome. Like, I throw my whole transcriptome on that, and so I think it took a couple of hours only on my computer, because I mean, it's they are interconnected, so it's very easy for the program to find the connections. And of course, then you have these huge, they are called hairballs. I don't know if you have heard about them. They are called hairballs networks, that you just have literally a hairball, and then you have to undo them. The other thing also that I think that is important, but I didn't have time to talk about, is that there are many softwares that allow you to play with your network once you have it. So I use Cytoscape. But Cytoscape, it sounds like just cells, but Cytoscape has, I follow them on Twitter, and there is a lot of like ecological networks. It has built to do that. Bye, Waitam.
I don't know if anyone else has any questions, but I've got one.
Yeah,
I have one.
yeah.
Just a little one. So I don't know anything about gene expression, so that was really interesting in the network analysis. But there's one slide where you mentioned gene ontology, and like the gene ontology gives like a lot of like very useless information or like confusing information that not necessarily give any meaningful result. So I've been using Revigo, I don't know how you say it, do you know it? To kind of like cluster all the gene ontology names together and kind of make it turn into a network of it. I just wonder what do you think about it?
Well, I have many opinions about gene ontology, but I think that I don't know with which organism you work with.
Non-model.
Non-model, so yeah. So that's the
no,
issue.
No
like,
reference genome, no reference transcriptome.
Yeah,
that kind of thing.
Yeah. So I think maybe Ira should be here because I mean I think it gives you very useful information at the beginning, because you are like, oh, this is like let me think one like cell stress response, like, oh,
What's
it's a
that
gene
mean?
that with cell stress response. Oh, yeah, that is cool. All these genes are related with cell stress response. And then you dive just a little bit deeper, and it's like such a over because gene ontology is like a family. I don't know if people know, but it works like a family tree, right? Like it goes the function of a gene. Let's say if it will cleave a protein, it will have like a tag, but then that protein that is, or let's say that we're working with a protein that cleaves a protein A that cleaves protein B. So protein B will be classified as a cleave protein that can cleave, I don't know, certain bonds, but it will also be classified as a stress response protein. So that like the gene ontology will tell you the two elements, that that protein cleaves a bond. It doesn't tell you anything, right? But that protein has a function in a stress response, then you get like excited. But actually that gene has been or that particular protein has been identified in certain model organism, and it can be that it has nothing to do with what the protein is doing in your organism. So that happens a lot in corals because they are little aliens, but I'm not sure. In vertebrates, it's much less, I think, because we are more related. So if the ontology is has and the functionality of that gene or protein has been found in rats, in humans, it might is more parsimoniously likely that it will do the same in your organism. So when you have all this information about gene ontology and about the function of your genes of proteins, I don't know what you work with, you have to be very careful not to overinterpret the data. And that's what a lot of people do. Like in the heat stress experiments are full of, like this type of overinterpretations of what genes of corals do, just because they use gene ontology labels. And I mean, I have done it as well. Like you have a resource and that's the only thing that you can use, but you have to be mindful that it might not actually respond to your requirements. Yeah. So the package that you use, what does it do? Like it kind of...
Yeah, it was kind of like, it's kind of like visualization tool of like gene ontology. So it groups like similar geoterms with similar with similarities together. So it gives you an overview of like cellular, like the main categorization categories of your geoterms, and then you visualize it on cytoscape. Cytoscape, yeah, too.
Yeah.
So yeah, I just thought like that's kind of related to a bit of network analysis, but also
it is network analysis. So all KEGG pathways have been done also with network analysis. I don't know if you know KEGG. Yeah. And Cytoscape is really good of like merging your data with other stuff, as you know. But yeah.
I mean, true. Just
exactly. Like be careful, because you can spend, like I spent months working on Cytoscape, months, but seriously months. And it's just so pretty, and it's like, oh, this can do this and this can do that. But then, yeah, it's all like real, like edges and relationships that need to be proven. So yeah. So it's nice to procrastinate on it though, because you build like beautiful networks and they look like so pretty. Yeah.
Thanks.
Yeah, no worries.
Any other quick questions? Just reaching the last minute or so of the meeting.
Yes. We need to know if you prefer 12 or 11. Now that we are 11. Who else? Anybody
11.
votes?
We're thinking about shifting the meeting time to 12, just because then it's coincides with lunchtime. People can, take an eating break. Some people are 1:11. If there's anything that majorly conflicts with it that we don't know, let us know. So we're seeing a lot of 11s. We'll probably do a poll on as well.
I mean, there's a sample bias here. We're all here at 11.
Yeah, exactly. We're talking about 2 weeks. It's survivorship bias.
Yeah. Survivorship bias. That's what you were talking about.
Yeah, this is a causal network, technically. When you get into it, you only examine the individuals that are here. You get an incomplete view of the total population. But yeah, we'll talk about that in 2 weeks. Do you have anything else to say, Natalia, before?
Not just that. Well, yeah, next week we don't have a session, but I guess that we can send an email around saying that we don't have a session. And then the next is you, Kevin, right?
Yes. I'll be talking about, a little bit here, causal networks. And what that means is just basically trying to infer when something causes something else in biology. Really difficult. It's never quite a clear line. There's a lot of things we don't know about, and those hidden paths can change how your entire statistical interpretation happens. So it's kind of really important to know, and something that isn't really taught a lot in stats. So I'm hoping to kind of share some illumination on that. Yeah. Anyway, that's in 2 weeks. So tune in.
Sally, I just remembered that I did a presentation about ontology and for my lab. Like, I know, were you there?
Yeah.
Where I like my title was like compromising is everything or something like that.
Yeah, I was there.
But I can send it to you if it might be useful.
Yes, please. Yeah, because I'm doing a lot of useless blast and
oh my god.
But everything goes back to flies, so I'm not sure that's very relevant to my octopus.
Yeah, sure, I can send it to you.
Thank you.
Can have a chat if you want.
Yeah.
Okay,
Natalia, cool. But are you planning to run another session with data, actually
Yeah,
analyze?
so the idea is to go through, because I have to use WGCNA. Sandra, you also can hear the drilling,
Yes.
right? We hate them. So yes, I have to use WGCNA. So I'm going to go through the tutorial, and maybe while I'm going through the tutorial, do the presentation and like have another session whenever we can, presenting the data. Mhm.
Okay.
Yeah.
Cool. Well,
Another-
thanks everyone for coming. And no session next week, but see you in 2 weeks. And then we'll be running the beginner course after that again. So we'll talk more about that. All right.
Okay.
Thank
See
you.
you. Thank you. Thank you for
Bye.
coming. Bye.
Bye. Thanks.